Hey guys, if you are new in my channel, please kindly click on the subscribe button to encourage me. Thank you.
All right, guys, let's continue our discussion on neurobiology or introduction to nervous system biology or physiology. In my last video, we have discussed neurons extensively, which is the structural and the functional unit of nervous system. I remember emphasizing that we have about 10 raised to power 11 neurons in our brain, and I remember calling our brain a bag of neurons because we have numerous neurons in our brain. And I remember saying that a neuron is made up of three parts: the axon, the dendrite, and the cell body. The axon, the dendrite, and the cell body. And I remember saying that axons are responsible for carrying information away from the cell body, while dendrites deliver information to the cell body. I remember saying that axons, they are wrapped within myelin sheath, which serves important functions. If you have not seen that video, I will advise that you check out for that video. It will really be helpful to you. And I remember talking about the cells that are called neuroglia. I remember telling you that neuroglia, they are support cells of the neurons. What they do is that they help the neurons to carry out their specific functions. And I remember emphasizing that the neuroglia of the central nervous system, they are different from the neuroglia of the peripheral nervous system. Four important neuroglia in the central nervous systems are astrocytes, oligodendrocytes, microglia, and ependymal cells, while the peripheral neuroglia are the swarm cells and the satellite cells. Note that the swarm cells, they are like the oligodendrocytes of the central nervous system. And a very quick recap. Remember, I said if you will not remember anything about astrocytes, note that they are the one responsible for the formation of the blood-brain barrier system. If you will not remember anything about oligodendrocytes, note oligodendrocytes secrete swarm cell, they secrete myelin sheath in the central nervous system, just the same way swarm cells synthesize myelin sheath in the peripheral nervous system. And when you hear the word microglia, what should come to your mind is the macrophage system. They are like white blood cells. They form the tissue macrophage system of the brain, while the ependymal cells are the epithelia that line the cavities and surfaces within the brain and the spinal cord. So you should always have that at the back of your mind. Remember, astrocytes, they create the framework for support and they form the blood-brain barrier system. Always remember that. And that will prevent injury to the central nervous system. And if there is injury, they are also the one that will stimulate formation of scar, meaning secondary intention of healing. Then you remember, I just talked about ependymal cells. You see, they are the one that line the cavities. So they line the fluid-filled cavities within the brain and the spinal cord. In the case of the brain, they will line the ventricles. I will soon make a video where I'm going to discuss extensively the ventricular system of the brain. And within the spinal cord, they line the spinal canal. And always remember that we have said that neurons can be classified based on various parameters. I will quickly do a quick recap before we look at that. But just before I do that, always remember that the cell body of neuron is just like the cytoplasm of your typical cells, meaning it's going to have nucleus. Inside the nucleus, we have the nucleolus. It's going to have mitochondria, which is going to supply energy for the neuronal activity. And we are going to have nuclear bodies, which is responsible for secretion of substances within the neurons. In fact, they are responsible. They are very important when it comes to production of neurotransmitters. Also note in this diagram that we have the axon arising from the point that is known as axon hillock. Note, this axon hillock is the only point that is devoid of Nisi granules, Nisi granules in the soma. And note that this axon, they will eventually terminate in skeletal muscle at the point that is called neuromuscular junction. I will watch out for my video on neuromuscular junction. Or they terminate on a specific site in another neuron, meaning they can communicate with muscle, delivering information to muscle, or they can deliver their information to another neuron. That point of communication between two neurons is what is called a synapse. Point of communication between two neurons is what is called a synapse. And some textbook or some authority will also add neuromuscular junction, which is the point of communication between the end plate or the terminal portion of an axon and a muscle fiber. We are going to discuss the synapse extensively in this video. You remember that we have said that neurons are responsible for transmission. The axons are responsible for carrying information from one neuron to another. Now, this information may be in form of electrical signaling or chemical signaling. We are going to discuss this extensively, and we are going to look at the differences between chemical synapse and electrical synapse. Remember, I've just emphasized that the point of communication between two neurons or neurons and muscle fiber is what we call a synapse. Now, this communication may be that they are sending electrical signals from one neuron to another, or they are sending chemical signals. So meaning we can as well say a synapse can be chemical or electrical. Now, also remember that we have said that we could classify neurons based on the number of processes that is coming out of the cell body. On that ground, we have the first group that are called multipolar neurons. Just the way it sounds, we are saying many poles are coming out of the cell body. We are saying many poles, like this example. In this, you have one axon and many dendrites, multiple dendrites and single axon. And this is the most common type of neurons. We find it all over the brain. Then we have the one that is called bipolar neurons. What makes them bipolar is the fact that you have one axon and one dendrite arising from the cell body. Don't ever forget that axons will always carry information away from the cell body, while dendrites will always deliver information to the cell body. A for away, D for delivery. All right. Then remember that these are not as common as the multipolar. But example of where we can find this is in our special senses. You know, when we say special sense, you know what we're talking about. The ear, the eye, and the nose. You have these bipolar neurons there. Then we have the least in terms of abundance, which are known as the unipolar neurons. They are not very common. In this case, you have just one projection from the cell body, which will now in turn divide. All right. So, also remember that we have said we can classify neurons based on functions by saying they are sensory neurons, they are motor neurons, or they are interneurons. What are sensory neurons? Sensory, just the way it sounds, they carry sensation from various parts of the body to the central nervous system for interpretation. Sensory, they carry sensation from different parts of the body to the central nervous system for interpretation. Immediately after interpretation has been done, then what will happen? The motor neuron will carry the instruction that is supposed to be done, will carry the order that is given from the central nervous system, motor, messenger. So now they carry the interpretation, the instruction given to various tissue and end organ for specific order for specific action. Yeah, for specific action. They will carry the interpreted information and the order that has been given to the end organ for specific action. Note that most motor neurons, they are actually multipolar. Now, most sensory neurons are either unipolar or bipolar. But for motor neurons, most of them, they are multipolar. This can be MCQ and you must take note. Most sensory neurons are either unipolar or bipolar in nature, but most motor neurons, they are multipolar in nature. Just the same way association neurons, which are also called interneurons, are also multipolar in nature. Do you understand that? So you must always have that at the back of your mind. Most multipolar neurons, they are either motor neurons or they are association neurons. You remember, association neurons are also interneurons. What do? They are the ones that are responsible for transmission of impulses between the motor and the sensory neuron. And they are the most common types of neurons, greater than 20 billion in the body. Also remember that anatomically, we may say neurons are neurons of the central nervous system, or neurons are neurons of the peripheral nervous system. And you remember that I said on my previous video that when you have collection of cell bodies within the central nervous system, you call them nuclei or you call them centers. When you have collection of cell bodies outside the central nervous system, you call them ganglia. Meaning collection of cell bodies within the central nervous system are called nuclei, while collection within the peripheral nervous system, or you say outside the central nervous system, are called ganglia. Of course, exception to this is the basal ganglia. Basal ganglia want to cause confusion. Basal ganglia is actually collection of cell bodies within the central nervous system. That's the only exception. So don't be confused. When you hear ganglia, they are actually collection of cell bodies outside the central nervous system. Don't allow basal ganglia to confuse you. Is that okay? All right. Then we also have, when you have bundles or collection of nerve fibers within the central nervous system, usually we refer to them as tract. And we are still going to talk about that when we talk about the tract within the central nervous system, pathway within the central nervous system. If you have not seen that video, watch out for it. Note that neural tissue are organized in specific manner within the central nervous system. This is the last thing I will talk about before quickly discussing synapse, which is what we are supposed to talk about now. All right, just like I stated earlier, synapse, the point at which nerve passes impulses from one to another. That point at which impulse or action potential passes from the presynaptic neuron to the postsynaptic neuron. Note, the aim of a synapse, or the purpose of a synapse, is to ensure that action potential passes from the presynaptic neuron, which is the neuron before the synapse, to the postsynaptic neuron, which is the neuron after the synapse. Note that the presynaptic neuron is the sending neuron. That is the neuron that is sending action potential to the postsynaptic neuron, which is the receiving neuron. The major purpose or the main purpose of a synapse is to ensure action potential move from one neuron to another. Note that a synapse is actually a junctional complex between the presynaptic membrane, which is the synaptic knob, and the postsynaptic membrane, which is the receptor surface for the incoming action, which is the receptor surface for the incoming information. If you are not going to remember anything about what a synapse is, remember the fact that is that point where action potential move from one neuron to the other. And you must note that that point is called a synapse. Then what is synaptic transmission? Synaptic transmission is that process by which neurons communicate with one another. That point is a synapse. The process is synaptic transmission. You must always have this at the back of your mind. The point at which action potential are transmitted from presynaptic to the postsynaptic is a synapse. The process by which that occurs is what is called synaptic transmission. There's something that you should always have at the back of a synapse. A synapse can be a chemical synapse or an electrical synapse. What is the difference? For chemical synapse, it involves the use of neurotransmitters that are released from the presynaptic neuron that will in turn bind to the receptor at the postsynaptic neuron, hence activating or initiating an action. What about an electrical synapse? Instead of release of neurotransmitter, it's just ions that will move and ensure continuity of impulse or action potential. Major difference between chemical and electrical is that one involves the use of a neurotransmitter, which is a chemical, while the other involves the use of electrical signal as a result of ion movement. We are going to discuss in details differences between a chemical and an electrical synapse. Let's do a quick comparison between chemical synapse and electrical synapse. One, a chemical synapse is a cell-to-cell junction through which nerve impulses are transmitted in one direction by means of neurotransmitter. Do you get that? One-way direction with use of neurotransmitter. What about an electrical? An electrical synapse is a cell junction between nerve cells through which a rapid transmission of nerve occurs by means of ions. Chemical uses neurotransmitters, which are chemicals. Electrical uses ions. That's one. Two, where do we find them? Chemical is found in higher vertebrate like human being. We have a lot of chemical. In fact, most synapses that are present in human, they are actually chemical, although we also have few electrical. But we have majority of electrical in lower vertebrates and invertebrate. Then what is the nature of the nerve impulse? Nerve impulse, we have said they transmit nerve impulse by releasing certain chemicals that we call neurotransmitters. While electrical, we have said they don't release neurotransmitter. Then how do they send information? Because there's a gap junction that allow direct communication between the two neurons. Get the difference, different from what I said initially. In this case, there's no direct contact in chemical. There's no direct contact between presynaptic membrane or presynaptic neuron and the postsynaptic neuron. Instead, neurotransmitters are released into the space between the two neurons called the synaptic cleft. Why in case of the electrical, there's direct contact and there's a gap junction that sounds like a bridge that connects the two neurons to each other, and this allows passage of ions from presynaptic membrane or presynaptic neuron to the postsynaptic neuron. Unlike the chemical where there's no direct communication, instead neurotransmitters are released into the synaptic cleft, which is the space between the two neurons. What about the direction of transmission? For chemical, transmission of signal occur in one way. It only occur in one way. The flow is in such that impulse move from presynaptic to postsynaptic. But in case of the electrical, the information is actually two-way. All right. Another thing you should note is that the size of the synapse, the chemical is larger, about 10 to 20 nanometer, while the electrical is smaller, about 0.2 nanometer. Another thing you should take note is that what is the content of the synaptic knob or the presynaptic membrane or the presynaptic or the presynaptic terminal? What is the nature? It is made up of the synaptic knob, that extension, is made up of synaptic vesicle and large number of mitochondria. Why? Because the process of production of neurotransmitter require energy. So the mitochondria is responsible for generation of energy, which will be used to synthesize the neurotransmitter. Now, what about the knobs of the electrical synapse? The knob lack synaptic vesicle, meaning they don't have synaptic vesicle and they have very few mitochondria. Why? Because they require less energy. All right, another thing we should take note is that at the postsynaptic membrane of the chemical synapse, they have chemoreceptors. What are chemoreceptors? They are the receptors that the neurotransmitters that is coming from the presynaptic membrane is going to bind to initiate specific action at the postsynaptic end. However, in this case, they don't need a postsynaptic receptors. They don't need a postsynaptic chemoreceptors. Why? Because there's no neurotransmitter that is binding to that. Instead, what they will have is just the ion channels that will allow inflow of ions, which will result in electrical transmission of impulse. Information transmission in chemical is slow. Why? Because action potential will need to arrive, neurotransmitter will be synthesized and released, neurotransmitter will now move and bind to the receptor at the postsynaptic end before they can initiate an action potential at the postsynaptic end. The steps involved will make the entire process to be slow, unlike the electrical, in which is just ion transfer. So that will make the process to be fast. Chemical, the transmission of impulse is slow. Electrical transmission of impulse is fast. The one that is slow is more vulnerable to fatigue. That's the thing. Whatever you do with at a very slow rate, the tendency that you get tired easily is higher. So electrical is less vulnerable to fatigue. Another thing is the one that require chemical synthesis, which are neurotransmitter, meaning the one that involve production or synthesis of neurotransmitter. They are also sensitive to other chemicals like hypoxia. pH, acidity, basicity can affect how they will function. But the electrical is insensitive to hypoxia or pH changes. Then where do we have the chemical? We find them in most junctions in the body of human. But where do we find the electrical in the body of human? We find them in sense organs and where we talk and where the brain is considered as a higher cerebral. We find them majorly in the special senses and where we consider as the site of higher intellectual function. So for these special senses, we find them in retina for sight, we find them in the olfactory bulb for smell, cerebral cortex, lateral vestibular nucleus, hippocampus. All these, the cerebral cortex, lateral vestibular nucleus, hippocampus, they are part of the brain that is very important in higher intellectual function. Then the next thing we are going to talk about is the process of transmission. Steps involved in synaptic transmission are as follows. Number one, we have arrival of action potential at the axon terminal. Number two, we have opening of the voltage gated calcium channels. Number three, then we have influx of calcium into the presynaptic neuron. Number four, we have calcium signal to the neurotransmitter vesicle. Number five, we have vesicles move towards the presynaptic membrane and docking will take place. Number six, we have release of neurotransmitter by exocytosis. Then number seven, we have binding of the neurotransmitter to the postsynaptic receptor. Then number eight, we have the signal initiation at the postsynaptic membrane, the effect of the neurotransmitter binding at the postsynaptic membrane. And finally, we have the inactivation of the neurotransmitter, which will end the entire process. All right, let's quickly look at them as fast as possible. Arrival of the action potential. Remember, we have said synaptic transmission is the process by which nerve impulses are transmitted from the presynaptic neuron to the postsynaptic neuron. So now these action potential that are being transmitted, they are the one that will first get to the axon terminal. Then on getting there, what are we going to notice? There will be opening of ion channel, voltage gated ion channel. Why is it called voltage gated ion channel? The reason is simple. Action potential is a voltage thing. So this is going to open the calcium channel. That's the step 2. Then the calcium will flow. Opening of the calcium will bring about calcium inflow into the presynaptic membrane. Influx of calcium will be followed by synaptic vesicle chemical interaction. The chemical interaction between the influx of calcium and the synaptic vesicle will lead to movement of the synaptic vesicle towards the end of the presynaptic membrane, which will now result in release of the neurotransmitter into the synaptic cleft. Now, the neurotransmitter will in turn bind to the receptors at the end of the postsynaptic membrane, the membrane of the postsynaptic neuron, and will now bring about a certain reaction that will now result in the transmission of impulse to the postsynaptic neuron. Now, after this, then there will be inactivation of the neurotransmitters so that will not result in fatigue. Note, if neurotransmitter was not inactivated, then fatigue will take place. We can summarize the 9th step or the 8th step into 4, which is 1, arrival of action potential at the axon terminal. You see the action potential arriving at the axon terminal, which is followed by opening of calcium channel. We can combine opening of calcium channel with influx of calcium into the presynaptic membrane. Then the number 3 will be the release of the neurotransmitters. How? Influx of calcium will initiate the process that will result in the binding of neurotransmitter to the presynaptic end membrane and release of neurotransmitter. Then finally, which is the 4th step, we have the binding of the neurotransmitter to the receptor at the postsynaptic neuron and activation of another action potential at the postsynaptic membrane, which will now result in transmission of action potential at that level, which will now be followed by termination of the action of the neurotransmitter via chemical inactivation. So that's another way to summarize. So you can now further break that down. Let's just remember acne. Number 1, acne, ACNE. A, arrival of action potential. C, calcium influx. N, neurotransmitter release. Then the E, event at the postsynaptic end, which is activation at the postsynaptic end. That's the best way to summarize. You can now further break each of those subheading down, so that will make it easy for you to remember. Finally, before we end this discussion, there are 4 criteria that must be met before we can classify a chemical substance as a neurotransmitter. Number 1, the molecule must be synthesized in the neuron. Number 2, the molecule must be present at the presynaptic neuron. And upon depolarization as a result of action potential, the molecule must be released into the synaptic cleft. Number 1, the molecule must be synthesized by the neuron. Number 2, the molecule must be present at the presynaptic end so that upon action potential that will result in depolarization, there will be release of this neurotransmitter. Number 3 is the fact that the exogenous form, like drugs that are having similar chemical structure with that neurotransmitter, if they should be administered, they must also elicit the same physiologic function. And number 4 is the fact that their action immediately after their action at the postsynaptic end, they must be inactivated. We must understand the mechanism of their inactivation. So now the criteria into 4S: source, site, synthetic form, specific termination mechanism. Source, site, Synthetic form, specific termination mechanism. What is source? We are saying before we can say something, a chemical substance is a neurotransmitter, it must be produced it must be produced in the neuron. It must be produced in the neuron. Then site, where are we supposed to find it? We must find it at the presynaptic end. The neurotransmitter must be present at the presynaptic end. Then the synthetic form must act like the endogenous form, and there must be specific termination mechanism that must be well understood. 4S that form the criteria for classifying a chemical substance as a neurotransmitter. Number one, source. The source must be from the neuron. Number two is site. We must find it at the presynaptic end. Number three is that the synthetic form must elicit the same physiology function as the endogenous form. Number four is the specific mechanism of termination that we must understand. When you remember those 4S, those form the criteria for classifying a chemical substance as a neurotransmitter. In my next video, we talk about action potential. I believe you enjoy the session. Thank you for watching this amazing video. I am so honored.